Okay, good evening, everybody, and welcome back. Today is going to be a continuation of the first micro and macromolecule we discussed last week. Remember, last week we discussed that we have four essential micro and macromolecules of life. We have the first one. What was that? What was the first micro and macro? They always come together. What was it? So, first of all, we had the proteins are the macromolecules. What are the micromolecules that make up these macromolecules? Amino acids. So we have amino acids. These are the building blocks for the macromolecules, the proteins. And then we had the monosaccharides and the disaccharides that are going to bind together to give us the polysaccharides. And we have the nucleotides that will give us different types of nucleic acids. And we have glycerol, and the free fatty acids, and together they give us the lipids. So today we're going to begin with the amino acids, and we're going to find out how they link up and how they become attached to one another to give us, as your friend nicely said last week, dipeptides, tripeptides, tetrapeptides, heptapeptides, hexapeptides, pentapeptides, and on and on. And we're going to learn today about the primary structure of a protein. What is a protein? A primary structure of a protein. What is the primary structure of a protein? What is the secondary structure of a protein? What is the tertiary structure of a protein? And what is the quaternary structure of proteins? We're going to survey a couple of very famous, very important, essential um, proteins that are required for your survival and for your reproduction. And we're going to focus on one of them. We're going to choose one of them, and that's hemoglobin. That is the most important protein. It's not the only one, not the only one, absolutely not the only protein, but it is the absolutely most significant protein. Okay, so let's not get ahead of ourselves, and let's begin by outlining that we have monomers. These are the building blocks. Building blocks are monomers. We also refer to monomers as micro molecules, and we build them up, we attach them to each other in order to get polymers. Don't let the words throw you off. Polymers or polysac oops, not polysaccharides. Polymers or macromolecules. Polymers or macromolecules. So we're going to begin today with amino acids. Amino acids belong in the bottom. Amino acids always belong at the bottom because we compile them and we polymerize the amino acids in order to get the very important polymers or the macromolecules known as proteins. So, yes. Yeah, you just have to find somewhere. Please just move up. You have to copy everything on the board. Or you can just, in the end, stay after class and copy it. Or you come up now. Move up a little bit. Yeah, you have to come up and you have to copy this. So please find somewhere. Please move up. Yeah, no problem. Okay, you're welcome. So we have monomers, micromolecules. An example of that is amino acids. It's not always going to be like that. So next week, we're going to replace this with monosaccharides. We're going to replace it with glycerol and free fatty acids. Sometimes we're going to replace it with the nucleotides. But today, our choice is proteins. So now, amino acids are going to be built up into proteins. And it's a highway. The highway is always two bidirectional, two ways. This direction now going from micromolecules to give me big macromolecules. This has a special name. What is the name of that? When you build up amino acids, micromolecules, in order to get a very big. Hmm? That's anabolism. Excellent. That is anabolism. So we have built up. And what's the usefulness of anabolism? Why do we always anabolize? We anabolize for storage. You build things up, you package things because you want to store them. You don't package things that you want to use, you package things that you want to store. So we package the monomers, the amino acids, in order to get the macromolecules, the polymers. Now, this is absolutely not a single direction, it is bidirectional. So now there is another direction coming down from very big molecules into very small molecules. What do we call that? Excellent. So this is catabolism. And catabolism, so if anabolism is for storage, we're storing energy. Catabolism is for breakdown and burning. Breakdown and burning of the energy. Excellent. And both words, there's a big umbrella to include them. Catabolism and anabolism, both of those. What do we name them? Yeah, that's metabolism. That's your metabolism. And every time we're going to have a different pathway. There is protein anabolism, protein catabolism. Carbohydrate anabolism, carbohydrate catabolism. Lipid metabolism, lipid catabolism. Nucleic acid catabolism, nucleic acid anabolism. You have to know all 8 of them. We're going to cover them. Are we clear? Okay. One thing that is very interesting about proteins is that you can never store proteins in the form of protein. You know, fat, you can store it in adipose tissue. You know, you store fats and lipids. You store them in your body in adipose tissue. We don't like the way it looks in our body, but that is how we store the fats. People who are suffering from obesity is because they're storing a lot of fat tissue, but they're not burning more than what they're storing. They're storing more than what they're burning. So we store fat in adipose tissue. We store carbohydrates, carbs, carbohydrates. We store carbohydrates in two places. In two places. In the muscle, and where else do you store your sugar? Your carbohydrates are stored where? Besides your muscle. Not the pancreas. The pancreas is important in the processes of the digestion. It helps the breakdown of your carbohydrates and also your proteins. We're going to learn today your nucleic acids and your lipids, but you don't store it there. Pancreas is too small to store any sugar. You store it in the, yes, in the liver. Excellent. So you store your carbohydrates in complex polysaccharides in the muscle and in the liver, and you store the fats and lipids in the adipose tissue. Do you store proteins somewhere in your body? No. You convert proteins. You convert proteins are converted into fats and stored in adipose tissue, or converted into sugars and stored in the liver and the muscle. This is really, really, really important. Is that clear? So, all the fat that is stored in your adipose tissue is not 100% fat. It's fat and is also used to be what? Proteins. Very good. And your sugar that's in your liver, and sometimes in the liver we have lipids also being stored there. But don't get fooled, it wasn't all the time sugar. Some of it used to also be proteins. So this is now our anabolism and catabolism. You need to know something. So when we are discussing anabolism, when we're discussing anabolism, we are talking about the different types of What is happening when we are performing anabolism? What is happening when we are performing catabolism? In catabolism and anabolism, we're dealing with bonds, with energy bonds. So in anabolism, no matter which micro or macro molecule are we dealing with amino acids? Are we dealing with proteins? Are we dealing with lipids? Are we dealing with carbohydrates? In anabolism, you are storing and putting energy in the bonds. So you're making a double bond, you're making a single bond. Sometimes you make a single bond. Sometimes you form a double bond. Sometimes you form a triple bond. Well, you're forming. So storing is about formation. Formation. Storage is about formation. We are forming bonds. Well, when we form bonds, we put energy in those bonds. So anabolism is about making macromolecules and storing energy in the bonds. Catabolism is not the same. Catabolism means that you are breaking the bond. You are breaking the bond. So the single bond is broken, the double bonds are broken, and the triple bonds are broken. So when you break the bond, so what's this business? Making bond, breaking bond, making bond, breaking bond, like the stock. So what is this making bond, break bond? Well, they realize that this is very, very important. When you form a bond, when you form a bond, the energy goes in. Some of you guys don't like the green color, but let's use black. So when you form a bond, the energy goes in. So the ATP, the energy is going to go in, or the free energy delta G plus plus, which means energy. We love energy, ATP, delta G. So when you make a bond, when you make a bond, the energy is in the bond because you are making a bond and you're storing the energy in the bond. When you make a double bond, a single bond, the energy delta G is always positive. Delta G is positive because the energy is put in the bond. When you make a triple bond, delta G is always positive. Positive means we have it. So your bank account is positive. It means there's money in your bank account. It means you're rich. So when you make a bond, it means that this molecule is rich in energy. That's why when we bind two amino acids, remember we said the amino acids always have an alpha carbon. Remember the structure of the amino acid. Amino acids always have an alpha carbon. Then they have a north, west, east, south. North, west, east, south. And we said that all of them, they share the hydrogen substituent. So all of them have the same hydrogen substituent. And they also share a carboxylic terminal. And they all have an amino terminal. We're going to look at that in the slides. Don't let that throw you off. We're going to look at it in the slides. This is just to make the point for now. And you have an amino terminal. You know, every amino acid, that's amino acid number 1. That's amino acid number 2. So when I have 2 amino acids, every amino acid has to have a hydrogen group. Check. Every amino acid has to have a hydrogen group. Check. Every amino acid has to have a carboxylic terminal. Check. Every amino acid has to have a carboxylic terminal. Let me put that one here. A carbonyl with an OH. Carbonyl with an OH. Every amino acid has to have an amino terminal right here. NH3, NH3. And the only difference is the R substituent. We'll talk more about that in the slides. The R substituent is what makes every amino acid different. So my point for now is that these are two amino acids. These are two separate micromolecules. I want to come back to this, storing the energy, the concept of storing energy. If I now, what did we say the synthesis reaction last week? If I want to make a bond, somebody gonna donate an H from this side, somebody's gonna donate an OH. What does that look like? An H and an OH is what? Water. So that's minus water, minus H2O. So water came out. What do we call this process? D. You see? Minus D. Dehydration. Synthesis. So we synthesize the bond. You see? That's the bond I was talking about here. When you make this bond, this is a very high energy bond. Is a rich bond. So when you made this bond through dehydration synthesis, this is the name of the process. Synthesis. And we saw last week, this happens all over. It happens in carbohydrates, it happens in lipids, it happens in proteins, and it happens in nucleic acids. You make a high energy bond, anabolism. You break the high energy bond, catabolism. So when you made the high energy bond, so now the energy went in the bond. So delta G is rich now. So now this molecule is a storage molecule now. So when I'm running the marathon, I break that bond, energy comes out. I feel a burst of energy. You see how the body works now? The body is all about making bonds so I can break them later. Anabolize, make bonds so I can break them down later when I need to exploit the energy. So you see here, I make bond for delta G so that when I break it, the energy comes out and I use it. Delta G comes out. So I store it and then it comes out later when I need it. I store it and then it comes out later when I need it. This is very, very, very important. Is that clear? This is not just happening in amino acids and proteins. This is happening all over. Monosaccharides is just the name is different. What's the name of this bond between two amino acids? What do we call that bond? That's a peptide bond. I store peptide bond, I store energy. I break peptide bond, I release energy. Between the two sugars, what's the name of that bond? Glycosidic bond. Excellent. I form a glycosidic bond, I store energy in the bank. I break a glycosidic bond, I release energy. I can use it when I'm running a marathon. I can build up energy in nucleic acid. That's called phosphodiester bond. I break phosphodiester bond if I want to release energy. I can build together a bond with glycerol and free fatty acids and so on. That's the business of metabolism. That's how we make energy. We're constantly shifting anabolism, catabolism based on your survival. Are you running for your life? You do catabolism. Are you relaxed on a Saturday night watching a movie and eating some food? Then you're not going to be burning it. You're going to be saving it. So we always save the energy. So we call this reaction of of storing the energy endergonic. Endergonic. N means in. I'm putting energy in. I'm depositing energy in the double bond. So when you go make a deposit, a check deposit or a cash deposit. Now when you make a bond in the protein, you make a single bond. That reaction is called endergonic. Delta G is positive. Delta G is positive. When I break down the bond, what's going to be the arrow, the direction of energy? When it was anabolism, it was endergonic, the arrow was pointing that the energy is being stored in the bond. Now, do you think when we break the bond, the energy arrow is going to point in or out? That's going to be pointing out, and this is going to be called exergonic. There is endergonic, there is exergonic. So in endergonic, delta G is positive. In exergonic, delta G is negative. So the energy came out because the bond was broken. Yes. No, it doesn't matter. It doesn't matter. But usually it comes in the front. So negative delta, because I see the negative, I know that I'm being now, the energy is coming out. I'm actually, do we like delta G minus or delta G plus? We like delta G minus because it means that the energy is coming out to me. So when I'm burning it. But when I'm just relaxed, the energy is going into the bond. So now the minus and plus is describing the state of the bond. So if the bond is positive, it means we're whole and we're storing it like you know the banknote, like a stack of cash. So it's just plus. But when you break it and you release the cash from it, so it's delta negative. That's exergonic. So yeah, this is a little confusing. One time I put it before, one time I put it after, but it's the same thing. So plus delta G, endergonic going in, exergonic coming out. This is an assessment assessing where the energy is. So now you, as a biochemist, looking at a reaction, you see positive delta G, you know the energy is going in a bond. You know that reaction is all about anabolism. That reaction is all about binding things together. But I never talked about the process of getting it out, and that is the thermal convention. So, how do I break bonds? Heat. So if I want to break a bond, I put heat in. And this is called endothermic. Endothermic. Thermal means heat. Endothermic. So I use I use heat to break my bond. So endothermic reaction, delta H represents the thermal convention. So when you see positive delta H, it means things are really hot and we're breaking a bond. Bonds, if you heat up, if you heat up water, it was ice. It was like 3 bonds. You heat it up, it becomes liquid, 2 bonds. You heat it up, it becomes gas, 1 bond. So endothermic, it means you put heat in. So if I want to save energy, oops, this is exothermic. You see, I got confused myself. This is exothermic because anabolism reactions are exothermic while catabolism reactions are endothermic. Endothermic. Because I don't break down in anabolism, I break down in catabolism. If I want to break down a bond, I put in. Endo means in. Endo means in. If I want to use ergonic, I'm talking about energy. If I say endergonic, that means the energy is going in the bond. If I say exergonic, that means the energy is exiting the bond. Are we clear? Endergonic means the energy is going in the bond. Exergonic means energy is leaving the bond. I'm catabolizing. Now let's go to a different convention. This is no longer ergonic, delta G. This is enthalpy. Enthalpy. Delta H, heat, thermal reaction. This bond that I'm going to store or this bond that I'm going to break, I'm going to need heat. So if I want to break down a bond, I'm going to heat it up. Am I going to use endothermic plus delta H? H means heat. Heat. If I want to break a bond, do I use plus delta H, or do I use minus delta H? Plus delta H. Plus delta H meaning that endothermic, meaning that the heat is going in the bond, is going to break the bond, the energy is going to come out the bond. You need to know the connection between the heat and the energy. So if I want to break a bond, I put in heat and energy comes out. If I want to make a bond, I remove heat. It's exothermic. Exothermic. I remove exo coming out. Exo minus coming out. So the heat comes out. Remember ice? I told you about ice. Ice is really solid. You heat it up, it becomes liquid. You heat it up, it becomes gas. So ice used to be triple bond. I heat it up, it became double bond. I heat it up some more, it became single bond. So when I did that, what happened to delta H? Did it increase or decrease? It increased because I was ice. I heat up ice. Then the bond was broken in ice. It became liquid. Then I heated up some more. Then it got broken. It turned into gas and it evaporated. But let's say I want to do the opposite. I freeze things up. What happens to delta H? Did delta H increase or decrease? It decreased because the heat now didn't go in the bond in the ice to break it. The heat now left the gas, turned it into liquid, and left the liquid, turned it into gas. Are we clear? So you need to develop an understanding between both endothermic and exergonic. They're always the opposite. They cannot be the same, pointing in the same direction. You can't put heat in the bond and energy in the bond. No. If you heat up the bond, you heat up ice, the energy is going to come out. Ice is going to melt. So the same thing. If you have a dipeptide, you heat it up, it's going to break. And once you break it, energy comes out. So you should see that all dehydration synthesis reactions are endergonic and exothermic. Thermic. And if I want to break the bond, that's called hydrolysis. Remember from last lecture, hydrolysis. I break down the bond. That's hydrolysis. I broke the bond. When the bond is broken, catabolism, is that endergonic or exergonic? Exergonic, because the energy is coming out to me. Don't let the endo-exo confuse you. Endo means toward, in the bond. Exo means coming out the bond. Ergonic means we talk about energy. Thermal means we talk about enthalpy and heat. Yes. So all dehydration reactions, you know that all dehydration reactions are synthesis, right? When I dehydrate, so I make a bond. So when I make a bond, is that endergonic or exergonic? That's endergonic, absolutely. And since I stored energy endergonic, what happened to the enthalpy, the heat? Did the heat also come in exothermic or did the heat come out endothermic? The heat exited. That is exothermic. Excellent. And we said the opposite is the lysis. 1 second. So hydrolysis, when I break the bond, whether I broke a peptide bond, whether I broke a glycosidic bond, whether I broke a glycerol and free fatty acid bond, whether I broke a phosphodiester bond, whenever I'm breaking a bond, the energy is always exergonic or endergonic. Exergonic, because I broke the bond, the energy is not going to be in a broken bond, it's going to leave the broken bond. And since I broke the bond with a exergonic reaction, what happened to the enthalpy, to the delta H, to the heat? Was it endothermic or exothermic? It was endothermic, absolutely. Is that clear? Is that clear? Okay. All right. So yeah, yes, yes. Enthalpy is heat. Enthalpy is another word for heat. So when you see delta H, that is the enthalpy. Positive delta H, that means it's endothermic. Heat went in. You need to know that. If I put for you negative delta G, I don't need to tell you that is exergonic. You know, negative means X. Whether it's in enthalpy, whether it's in endergonic or exergonic reactions. Minus, you immediately know that something is coming out. Something is coming out. What is coming out? Minus delta G, energy is coming out, exergonic. Minus delta H, heat is coming out. The enthalpy is being reduced. So that's an exothermic reaction. Yeah. The exo, so let's look at it. The exo, so the exothermic means that the energy came out. The heat came out, not the heat energy came out. Yeah, so negative delta H. So when the heat is being removed from the bond, remember ice. If the heat is being removed from a liquid, it's gonna freeze up. So that is gonna be endergonic because the energy is now gonna be stored. It's gonna be built. Endergonic, the energy is gonna be built. Look at here. When you put heat, when heat moves out, when it's an exergonic reaction, when heat moves out, the bond forms. Now there is energy in the bond. That's endergonic. Okay. Yeah, building anything, building proteins, building carbohydrates, building nucleic acids, building lipids. So there always have to be the opposite. You can never have an endothermic and an endergonic reaction at once. Okay? So what's your question? you guys are you guys cannot ask the question. What's the question? So the question is, look here. If you make a big protein of multiple amino acids, you're making bonds or you're breaking bonds. If you're making a protein, are you making bonds or you're breaking bonds? You're making bonds. So in order, when you're making bonds, you're making energy or you're losing energy. You're breaking energy. Yeah, you're making energy. You're storing energy. Energy is going in. You're saving energy. So when you make a fat tissue, you're storing energy. But when you run the marathon, you break down that energy in order to exploit it for for survival. Okay? Though we're gonna go over it more in the slides. Okay, so now today our main topic is amino acids and the proteins. Proteins, we talked last week that they are very important in the sense that these proteins are important because they are receptors, extracellular receptors. Remember the example I gave you about how if you have an Uber ride, they cannot come into school, they have to talk to a second messenger. The outside receptor is a protein. The intracellular receptor, the intracellular part of the receptor is a protein. We talked about cyclic AMP, which is a nucleotide. That's a second messenger. But in addition to the second messengers, we have other enzymes, which are proteins. So proteins are everywhere. They are receptors, they are protein channels, they are structural proteins like collagen, they are like enzymes, which is our main topic for today, is the hemoglobin. So now, yes, hemoglobin. Yes, hemoglobin. So, now we talked about saving energy, storing energy, and now we need to talk about if you pair up, make these peptide bonds, if you pair up these peptide bonds together, you're going to have a linear chain of amino acids. That's called a primary primary structure. That is called a primary structure of amino acids. This primary structure, remember we said every amino acid, they have 3 out of 4 things the same. No matter which amino acid are you're going to have 3 out of 4 that are identical. The H substituent, the carboxylic terminal, and the amino terminal. The only different part that makes all the amino acids unique and different from each other is the R substituent. So this R substituent is what defines the amino acid. There are polar amino acids because the R substituent is polar. There are non-polar amino acids because the R substituent is non-polar. There are aromatic substituents, because aromatic types of amino acids, because the R substituent is aromatic. So if there is anything special about a particular amino acids, it is always the R substituent that is causing this particular property for the amino acids. So these amino acids, that's amino acid number 1, number 2, number 3, number 4, number 5, number 6, they're not going to stay there in solution. 1 is going to start to have an interaction with 2, 2 is going to have an interaction with 3, 3 is going to have an interaction with 4, 4 is going to have an interaction with 5, 5 is going to have an interaction with 6. What is that interaction based on? The R substituent, the R group that makes the amino acid different. Then the primary structure is going to fold and give us the secondary structure. The secondary structure. And we have 2 types of secondary structure. An alpha helix, and the second one is the beta pleated sheets. The alpha helix and the beta pleated sheets. This is a secondary structure. We don't stop here. We move on into a third tertiary structure. This third tertiary structure is going to form for us through disulfide bridges, and salt bridges. It's going to coil up to give us a tertiary structure protein. The most famous type of tertiary structure protein is collagen. So collagen is a protein that operates perfectly when it's tertiary. And then there are peptide interactions that are going to give us number 4, quaternary structure. There are quaternary structures. This quaternary structure is an example of that is our good friend hemoglobin that we will discuss today. So in quaternary structure, you have different polypeptides, subunits that are going to be joined together to give you a conglomerate, to give you a very big protein that is made up of little companies, that is made up of little different peptides, different peptides coming together. As we will see with hemoglobin, is made up of 4 different subunits, 2 alpha and 2 beta. That's for adult hemoglobin. For babies, it's fetal hemoglobin. It's made up of 2 alpha and 2 gamma. We will get to that. But what you need to know for now is that we always come at the ribosome like this, like a long linear polypeptide. Primary structure, long linear polypeptide. Long linear polypeptide structure coming at the ribosome. Then that primary structure is going to fold due to what? The folding that gives us the alpha helix and the beta pleated sheets. Due to the R substituent properties. Is it a polar group? Is it a non-polar group? Is it aromatic? Does it have a sulfur bond to make a cysteine or disulfide bridge? So your primary structure is very unique. And this brings us to the Christian-Anfinsen experiment. This is very important. Christian-Anfinsen experiment. This was a Nobel Prize. This was a Nobel Prize winning experiment. And it's a very intuitive, very interesting, and very intuitive experiment. What he did was, this scientist, Christian Anfinsen, they all believed that the most important structure is the quaternary structure because it's the most complex. So what he did was, when you go from primary structure to secondary structure to tertiary structure to quaternary structure, those bonds that are made, this is called folding. We're folding. We're folding from the primary to the secondary. We're folding from secondary to tertiary. We're folding from tertiary to quaternary. Now, this folding is based on what types of bonds? We talked about them last week, those dashed bonds. Yeah, intermolecular bonds, hydrogen bonds. Very good. Those are not covalent bonds. No R substituent between 1 and 2 is going to form a covalent bond and go and share electrons between amino acid 1 and 2. No sharing here. It's just like a dipole. It's called dipole moment. Dipole, 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 dipole moment, where there's one negative charge and one positive charge. Remember how oxygen is so big and it has like an inherent negative charge, and some hydrogen has like a little minimal positive charge, and then they bind together. Remember that? So that's all the folding is about. Folding is non-covalent. You need to really know that. Folding is not really covalent. Folding is like hydrogen bond. It's like a weak bond. Remember, I told you last week, you have classmates in one class. Next semester, they're not there with you anymore. The next semester, they are with you now. So it's like a broken and a made type of bond. Covalent bond, that one never breaks, at least not easily. So that's why if you have a protein in a tertiary structure, if you heat it up, all the hydrogen bonds break and you go back into the primary structure. If you are a secondary structure, you heat it up. So heat is going to reverse everything up. Look at the direction. The direction here was folding, 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 folding. But if you apply temperature, if you increase heat, if you add delta H, endothermic, if you heat up a protein, it's going to start to denature. You need to know this word, denature. That's a unique word for protein. That we had a protein that used to be quaternary, now it folds, rolls back, it becomes tertiary, it falls back, it becomes secondary, it falls back, it becomes primary. Are we clear? So heat is going to cause denaturation. Is it only heat? How about if you mess up the acidity? If you put a protein in like some acidic environment or some basic environment, is it still going to stay in a quaternary tertiary structure, or is it going to denature? You really need to know that. Why are we denaturing? Because the folding bond was a hydrogen bond, was a salt bridge, disulfide bond. salt bridge, disulfide bond, hydrogen bond. Those are bonds, but they're like a, like a neighbor. You change your neighborhood. That's like a class. You change your classmates. So all of that is susceptible to heat. But once you're back into the primary structure, can heat and salt and pH break down the bond in the polypeptide? No, those ones are covalent. Those ones don't break. I mean, unless the heat is like volcano or like the acidity is like insane, then they will break. It's too much. But I'm talking about going from the the tertiary to secondary. So the proteins are constantly going to denature. So that brings us to Christian again, Christian Amphenson's experiment. What he did was he brought a protein in a quaternary structure. He brought a protein in a tertiary structure. Then he heated up, he added heat to it. Then he started to add acid and he messed up the pH. What do you think happened to this quaternary structure protein? What do you think happened to this tertiary? It what? It, say it. I cannot hear you. Say it. It defolded, but we say the word I like defolded too. They said no. I used to like back when I was in college. They said defolded. The professor said no, denature. So, anyways, but at least you know it denatured. But what led him to get the Nobel Prize was after it denatured, he removed the heat, so he cooled it down, gave it normal temperature. Then he removed all the crazy pH, and he called it like he gave it like a normal pH, and he realized that this protein rolled right back and folded into its accurate function. So, for example, collagen. We all know collagen is tertiary. Or myoglobin. You know myoglobin? Oops, I said collagen. Oh, collagen is secondary. Oh no, collagen is secondary. Collagen is a secondary protein. Myoglobin is tertiary. I'm sorry. Myoglobin is tertiary. So he did that. And what's an example of a quaternary protein? Hemoglobin. Hemoglobin is quaternary structure. What's going on? Hemoglobin. Okay, hemoglobin. Hemoglobin. Okay, hemoglobin is quaternary. Okay, hemoglobin is quaternary structure. Myoglobin is tertiary structure. Collagen is secondary. Keratin is secondary. So he brought all of them. He knows them. Hemoglobin is fourth. Collagen is 2, and myoglobin is 3. He knows them. He put them in different beakers, and he put acid, and he heated up, and he saw that all of them, no matter who you are, if you're hemoglobin, you go back to primary structure. If you are collagen, you go back to primary structure. If you are keratin or collagen, you go back to primary structure. Then he came to all of them, cooled them down, removed the crazy heat, and he removed all of the pH disturbance and restored the isoelectric point. And he saw that all of them returned back to their actual original structure after they rolled all the way back, denatured, defolded, denatured all the way to the primary structure. Then he concluded that the most important structure is not the quaternary structure, is not the tertiary structure, is not the secondary structure. It is the primary structure, and he called it the transcript. That's the one that has a transcript of how you're supposed to look like. That's like the DNA. Your DNA tells you is gonna say, how are you gonna look like when you're born? The primary structure is the transcript that tells you, is collagen going to become tertiary or secondary or quaternary? Is hemoglobin going to be tertiary, quaternary, or secondary? Because that primary sequence has the R group. It has the R substituent group. Are we clear? And we're going to learn today that, based on this order group, there's many different conventions. Some even nutritionists are classifying amino acids now. Some amino acids are essential. Some amino acids are non-essential. Some amino acids are essential, means you need to get them from diets. And some of them are non-essential, meaning that your own body can make them. If you're not sick, some non-essential, your body can make them, but if you're sick, you can't even make them. Yeah. So students think that because an amino acid is non-essential, you don't need to eat it. No, you take it into account that your patient is not sick. Because if you're sick, even you're non-essential, you need to take them from diet. Are we clear? So the last thing we go before we go into the slides is, you know, there is different types of proteins. You guys took A and P. Actin is important, like a cytoskeletal protein. Myosin is important for the contraction. Different types of proteins. We have actin, collagen. This collagen is serious. When you take pathophysiology, you will learn about Ehlers-Danlos disease. Anybody heard of Ehlers? Ehlers, Ehlers-Danlos disease. Ehlers-Danlos disease is a disease where there is a genetic problem. They can't make collagen. Those patients, they have a genetic problem, so the gene that makes collagen is defected and mutated, is not working. So when you transcribe that gene, you're never getting collagen. So they have a connective tissue problem, and their joints are so weak and wobbly, they're like dancing, like Korea all the time. Why? Because they don't have that structural tensile integrity that they get from collagen. Hmm? Celine Dion from Titanic? I don't know what she has ever since she said some statements. Anyways, so, so, but you need to. Our point is, if you damage or defect a particular gene, you cannot make the protein, because we already discussed last week in the central dogma of biology. You have a gene, you transcribe the gene, you get a protein. Hemoglobin, it doesn't come from the heaven, it comes from the gene. So you transcribe the gene. That's why in sickle cell anemia, that's a problem. Why? Because in order to transcribe the gene for hemoglobin, there's a mutation. So now instead of having glutamic acid, we have valine. Who said that? Very good. Excellent. Very good. So now the cells will sickle, because you replace one that's not supposed to be there. You're supposed to have glutamic acid, but now you have valine. So this is a genetic problem. The ribosome is going to say, listen, give me the right codon, I'll give you glutamic acid. The polymerase is going to say, I can't find it because there's a mutation. Are we clear? Okay, so we're going to find out today that this hemoglobin is very important, because it's responsible for it's responsible. We know what I mean. Hemoglobin, you know what I mean. So you know that when I ask students, what is the function of hemoglobin, they say, well, it transports oxygen. What about carbon dioxide? Well, you can't say that. You can't say, because if you say that hemoglobin is transporting oxygen, it means that it's not doing the downtown trip. Every subway, it has an uptown trip and a downtown trip. So we're gonna learn today that there are two trips, uptown and a downtown trip. Yes. Wait, what? Okay, hurry up, because we need to move on. Okay, that's why I said make a group, cooperate with each other, and send these and prompt each other and cooperate on doing the good. Don't cooperate doing evil. and work together and help each other out. Hmm? Okay. You don't need to take a photo. You need to copy it down. That's all. Okay. It's okay. We're gonna make it work. Let's do our best. Patrice Keem. I remember you from Biolab. Shaquille too. She was with Shaquille. Oh no. Shaquille is here today. Shaquille, my friend. Oh, Shaquille. Shaquille is my best friend. What are you talking about? Oh my God, Shaquille gave me nightmare in Biolab. Oh, I was gonna retire. Okay, okay, I'm not going too far. All right, so let's maximize the time. What time is it now? 6:20. All righty. So I think we have just the right time before we go on break and come back to the slides. Let's do the uptown trip and the downtown trip of hemoglobin. So that when we do the slides, you guys remember, oh, I remember that. I copied that down from the whiteboard. Oh, I remember that. Okay. ALS? What are you guys talking about? I'm sorry. Can I just put this here? Okay. Are we ready? I hit her MacBook. Okay, alrighty. So let's talk about our uptown trip and downtown trip. So at the top, you have your lungs. Those are your lung. This is your lung, in case you can't tell. This is the lung. And you have here what is that? What is this here? Shh. This is really important. Guys. What is this? No, this is tissue. Absolutely, that's tissue. That's all we need, tissue. Because hemoglobin, don't say adipose tissue, because every tissue needs oxygen. Hemoglobin goes everywhere. Hemoglobin goes to nervous tissue, epithelial tissue, bone tissue, connective tissue, muscular tissue, you name it, hemoglobin is going there. So we're just going to say tissue. So there's two trips. A trip coming down to the tissue and a trip going up to the tissue. You see now from A and P, you know that red blood is like rich in oxygen, is going to be rich in oxygen. Oops. Red blood is going to be rich in oxygen because the partial pressure of oxygen in the lung is very high, because you just took a deep breath. So your lung has a high partial pressure. The partial pressure of oxygen in the lung, is it high or low? High, because your lung is your reservoir of oxygen. So the partial pressure for oxygen in the lung is very high. What do you think the partial pressure of oxygen in your tissue, who is burning all of the catabolism and all of the breaking down of the carbohydrate catabolism, and protein catabolism, and nucleic acid catabolism, and lipid catabolism? You think something like that is going to be rich in oxygen? No. So the partial pressure, PO2, the partial pressure of oxygen in the tissue is going to be very low. The partial pressure of oxygen in the tissue compartment is very low. And the partial pressure of oxygen in the lung compartment is very high. Are we clear? Now, in your lung, what do you think the partial pressure of carbon dioxide is in your lung? Is your lung full of carbon dioxide? No. Your partial pressure of carbon dioxide in the lung is so low. What about your tissue? Your tissue is constantly burning all of the toxic waste and the metabolic waste. Do you think this tissue is going to have high carbon dioxide partial pressure or low carbon dioxide partial pressure? Very high. So your tissue is going to have extremely high, very high carbon dioxide partial pressure. This is really, really, really important. This is the mental picture that you should have about the two trips before. This is station number one, that's station number two. Obviously, we're ignoring the heart and we're ignoring all the intermediates, but that's the ultimate. That's station number one, that's final station. So my lung is full of oxygen and has no carbon dioxide. My tissue is burning because it's making me walk and is performing a lot of survival and digestion and reproduction and locomotion, so it's gonna have no oxygen. Why does the tissue have no oxygen? Because it's burning all of it. Remember oxidative phosphorylation, aerobic respiration? Well, why they call it aerobic? Because it uses oxygen. Why they call it oxidative? Because it uses oxygen. So it burns oxygen. And when you burn something, your car, when you burn the gasoline, all the fume gas coming out. So all the waste carbon dioxide is going to be very high in the tissue. But the lung is going to have no carbon dioxide because it's rich in oxygen. So now your hemoglobin is going to do both. Your hemoglobin is going to have one trip where it's gonna go downtown, where hemoglobin, this is your H, hemoglobin, is gonna be rich in oxygen and poor in carbon dioxide. That's our downtown trip. Our downtown trip is we coming from the lung. We are full of oxygen. We can't wait to get to the tissue so we can give the tissue the oxygen for oxidative phosphorylation. I'm sure somebody's gonna forget it. Oxidative phosphorylation, phosphorylation, and aerobic, aerobic respiration. Oxidative phosphorylation and aerobic respiration. And when we perform it, we consume all the oxygen in oxidative phosphorylation. We consume all the oxygen in aerobic respiration. And now we have nothing but the waste carbon dioxide. Then that's the trip number two for hemoglobin. That's the uptown trip. So now the oxygen is gonna be so low and the carbon dioxide is gonna be so high. Isn't that interesting? The same hemoglobin. One time is full of oxygen, no carbon dioxide. The next time is full of carbon dioxide and no oxygen. Once you bring in high oxygen to the tissue, we're gonna burn it. We're gonna burn and use all of the oxygen and give the waste. And we're gonna go up here. The lung is gonna take all the waste, carbon dioxide, and throw it out and take in oxygen. And that's how it's going. Well, today you're gonna learn that hemoglobin does both. That's why students, what does hemoglobin do? It carries oxygen. It carries oxygen from the lung to the tissue and carbon dioxide from the tissue back to the lung. So you got to give it its two trips, uptown and downtown. So you're going to learn today that there are factors that affect this binding of oxygen. Number one, there is cooperative binding. What is cooperative binding? Cooperative binding means once hemoglobin gets to its destination, there is a positive feedback. Positive feedback. What does positive feedback mean? It means that, let's look at this trip here, going from the tissue to the lungs. When you go from the tissue to the lung, you have so much carbon dioxide. The moment you get to the lung, if one carbon dioxide come out, then number 2 right after it, number 3 right after it, number 4 right after it, there is a positive feedback cooperative activity. So if one is released, the other one's right behind. They're all followers. It's like a down slippery slide. Same thing with the oxygen. If one oxygen bind from the lung to the hemoglobin, then the second one is going to bind faster, then the third one is going to bind faster, then the fourth one is going to bind faster. So they affect each other. That's cooperative binding, positive feedback. If one binds, it's not going to interfere with the others. Don't think that if one oxygen binds, the other oxygens are not going to have a seat. They're not like human beings in the subway. They don't cooperatively sit with each other. They do. If one oxygen bind, 10 will right after it. They are so happy to make space for each other. Not only in getting on the ride on hemoglobin, but also leaving also. If they want to leave into the tissue, if one leaves, the other ones are so eager to leave with it. That's for oxygen. What about carbon dioxide? If hemoglobin takes carbon dioxide, then carbon dioxide 1 jumps in, then 2 follow it, then 3 will follow it, then 4 will follow it, and so on. And once we get to the lung, if they want to leave from hemoglobin into the lung, if one leaves, the others. So it's cooperative loading and unloading of carbon dioxide and oxygen. Positive feedback, cooperative loading of oxygen and carbon dioxide, and cooperative unloading of oxygen and carbon dioxide. Can you have one trip where both are in the same hemoglobin? Can you have one hemoglobin that has both oxygen and carbon dioxide at the same time? No. That's like you go on an up-down trip and you have like somebody who's going downtown at the same time with you. It doesn't make sense. All the hemoglobin going downtown, they are full of oxygen. Why would there be carbon dioxide? Why would there be carbon dioxide on a trip downtown to tissue that needs oxygen? And why would there be oxygen in a trip where we're going where there is low partial pressure for carbon dioxide? And you know the movement is always from high to low. So the hemoglobin is going from high partial pressure of oxygen to low partial pressure of oxygen. From high partial pressure of carbon dioxide to low partial pressure of carbon dioxide. So did we get cooperative binding, positive feedback, cooperative loading and unloading of carbon dioxide and oxygen out the way now? Okay. And the last thing is the Bohr's effect. So the Bohr's effect is a very interesting type of a graph. So in Bohr's effect, we gonna learn in Bohr's effect that as a function, Wes, what's going on? In Bohr's effect, we're gonna have a graph like this, x-axis and a y-axis. On y-axis, you're going to have the partial pressure of oxygen. On the x-axis, you're going to have some like stressful thing. Like for example, some stressful thing would be carbon dioxide. Some stressful thing would be heat, it's too hot. Some stressful thing would be acidity, like somebody suffering from, acidosis. You think somebody who's suffering from acidosis, their hemoglobin is going to perform the same way as somebody who's healthy? So absolutely, stress, Bohr's effect, Bohr's graph is going to teach us one valuable lesson. Bohr's graph is going to teach us a valuable lesson. Bohr graph is going to teach us the following: that if naturally hemoglobin has a sigmoid curve like this, meaning if this is the partial pressure of oxygen, 10%, 20%. Partial pressure means how much oxygen is there. A place with high partial pressure of oxygen, it means there's a lot of oxygen. A place with a low partial pressure of oxygen, it means there's no oxygen. So partial pressure means how much oxygen is there. 30% oxygen, 40% oxygen, 50% oxygen, 60% oxygen, 70% oxygen, 80% oxygen, 90% oxygen, 100% oxygen. So this graph is showing you that normally this shows you partial pressure of oxygen, and this here will show you the saturation of hemoglobin, the saturation of hemoglobin with oxygen. Now, I want to ask you a question, just an intuitive question. Don't get too paranoid with copying. Think about it this way. Let's say there is a hemoglobin that is rich in oxygen, and it came by two tissues. One tissue is suffocating. It has only partial pressure of 10% oxygen. It has only 10% oxygen. Then it passed by another tissue that has 30% oxygen. Do you think hemoglobin is going to give the same amount? Is going to give the same amount of oxygen to somebody who has only 10% and somebody who has only 30? Who is it going to give more to the temper? Absolutely. So Bohr discovered that hemoglobin is not gonna work like equally. Where there is more problem, is gonna unload more. Problems could be partial pressure. For example, if I'm running on a treadmill, who's gonna have a low partial pressure of oxygen? My digestive system or my musculoskeletal system? My musculoskeletal system. So when hemoglobin passes by my intestine, it's going to give a little bit of oxygen. But when it passes by my thighs and my musculoskeletal system, it's going to unload more. That's just about partial. What about heat? What about heat? What if things, what if you just, like in this room right now, your hemoglobin is working. What if we increase the heat and it's really hot now? Do you think hemoglobin is going to be giving out the same oxygen to your tissue as when it was cool? It's going to give more oxygen. So that's the second is stressor. First stressor was the amount of oxygen. The less oxygen, I give you more oxygen as hemoglobin. How about heat? If you're too hot, I give you more oxygen. Hemoglobin is your friend. The more you need, the more it gives you. How about acidity? If you have acidosis, it's going to give you more oxygen. So Bohr discovered that this sigmoidal curve is going to shift, is going to shift right, is going to shift right, is going to shift and move to the right. What does that mean? Shift to the right. Hmm? It means that we accommodate. We accommodate. This Bohr graph, this sigmoidal curve, Bohr's graph, is not set in stone. It's set in stone when you're homeostatic, healthy, sitting down, watching a movie, all relaxed. But once you're under stress, it's not going to be the same. It's going to shift to the right, meaning that it's going to be accommodating you. So you're going to see that the less partial pressure of oxygen in the tissue, the less oxygen will be on the hemoglobin because it gave all of it to you. Let's say I'm hemoglobin. I have 100% oxygen. I pass by a tissue that is 10% oxygen. I'm going to give it 90% of what I have. So remember that. The lower the partial pressure, the lower the saturation of hemoglobin with oxygen. If hemoglobin, okay, so the lower the partial pressure of a particular tissue with oxygen,